I have a PhD in wildlife biology. I have spent 17 years studying animal behavior, tracking patterns, identifying species from partial prints, partial scat, partial remains. I have published 11 peer-reviewed papers. I have taught graduate-level courses on predator behavior in closed ecosystems. I am telling you this because I need you to understand what it means when I say I could not identify what I was tracking. Not that it was rare. Not that it was unusual. Not that it was a species I had not personally encountered before. I mean, I could not place it in any known family, any known genus, any known order of animal life currently documented on this planet. I tracked it for 7 years. I have 4,000 hours of field notes, photographs, plaster casts of prints, audio recordings, and biological samples. Every single sample came back inconclusive from 3 separate laboratories. Not unidentified. Inconclusive. There is a difference. Unidentified means we know what category it belongs to but cannot name it yet. Inconclusive means the sample did not match the biological markers of any known category of life on Earth. 3 labs. Same result. Every time. My department head told me to close the study 14 months ago. He said the research was, and I am quoting him directly, producing results that are not useful to anyone. I asked him what that meant. He said, "It means stop, Sarah." First time in 7 years he had used my first name. I did not stop. 2 weeks later my entire field research database was wiped from the university server. I had offline backups. I still have everything. My name is Dr. Sarah Okafor. And what I found in the Appalachian forest does not have a name yet. But it knows mine. It started with a print. Standard procedure on day 1 of any field study. Walk the grid. Catalog what is there. Establish baseline species presence before you start looking for anything specific. I was cataloging in the northern section of the study zone, dense old growth, minimal human traffic, exactly the kind of terrain where you find clean undisturbed animal sign. The print was in soft mud beside a creek bed. At first glance it looked like a large canid. Wolf-like. 5 toes, strong claw impressions, deep heel pad. But the proportions were wrong. The stride length between prints was consistent with an animal roughly the size of a black bear. But the print itself was the size of a large dog. An animal with a bear's stride and a dog's foot size would have a skeletal structure that does not exist in nature. I photographed it. Cast it in plaster. Sent the cast to my department. My department head examined it and said it was probably a malformed wolf print. Injury or congenital defect distorting the normal impression. I accepted that explanation. For about 3 weeks. Then I found a second print. Different location. 20 miles north. Identical proportions. Identical distortion. Identical impossible stride length. Same animal. Different territory. A malformed individual does not cover 20 miles of mountain terrain. I expanded my study grid. Over the next 2 years I found 43 separate print sites across a 200-mile stretch of the Appalachian range. Not 43 prints. 43 separate sites, each containing multiple prints showing the same animal moving through at different times of year. The distribution was not random. I mapped every site and the pattern that emerged was not a territory. Not a migration route. Not a feeding circuit. It was a perimeter. Whatever was making those prints was not moving through the forest randomly. It was moving around something. Consistently. Deliberately. Maintaining a boundary around a specific central zone of approximately 40 square miles that sat in the middle of my study area. I had been working inside that zone for 2 years without knowing it. The animal had been circling me the entire time. I set up cameras. 47 cameras across the perimeter line. Motion triggered. Running continuously. In 14 months of footage across 47 cameras, I captured exactly 0 images of the animal making those prints. But on 63 separate occasions the cameras captured something else. The moment just after the animal had passed. Bent grass. Disturbed leaves. A branch still moving. Mud still settling in a fresh print. 63 times it passed within feet of my cameras. 63 times it avoided the frame by seconds. It was not avoiding my cameras by chance. It knew exactly where every single one was. In year 5 I got lucky. A fresh kill site. Whatever had made those prints had taken a deer sometime in the previous 6 hours. Clean kill. Precise. No signs of struggle beyond the initial takedown. The carcass had been partially consumed in a way that matched no known predator feeding pattern I had ever documented. But there was biological material at the site. Saliva on the carcass. A small amount of what appeared to be blood on a broken branch nearby. Not deer blood. Different color. Different viscosity. Almost too dark. I collected everything. Packaged it properly. Sent it to the university lab under a neutral sample code without identifying the study it came from. 3 weeks later the results came back. The lab flagged the sample immediately. Called me directly rather than sending a written report. The technician said, "Dr. Okafor, where did this sample come from?" I asked why. She said, "Because the cellular structure is not consistent with any animal in our reference database. The DNA sequencing keeps returning errors. Not gaps. Not unknowns. Errors. Like the sequencer cannot process what it is reading." I sent the same sample to 2 more labs under different codes. Same result. Both called me directly. Both asked where the sample came from. Neither sent a written report. No documentation required. I heard that phrase before. In a story a former ranger named Aaron Cross once told me at a conference 3 years before I understood what it meant. In 7 years of field work I had never once felt watched. I had felt followed. I had felt assessed. I had occasionally felt the specific quality of silence that means something large is nearby and choosing not to move. But watched is different. Watched means something is making a decision about you specifically. On a Tuesday morning in October of last year I was sitting at my field station reviewing camera footage. I felt watched. I looked up from my laptop. 30 feet away at the tree line stood something I am going to describe as carefully and accurately as I can. It was standing upright. Roughly the height of a large man, but the proportions were not human. Shoulders too wide. Arms hanging at a length that was slightly wrong. Head tilted at an angle that suggested it was examining me the same way I had spent 7 years examining everything in that forest. It was completely still. It stood there for 11 seconds. I counted. Then it stepped back into the tree line and was gone without a sound. No rustle. No crack. No displacement of undergrowth. Just gone. I looked down at my laptop. On the camera feed from camera 31, the camera closest to where it had been standing, the footage showed nothing. Clear tree line. No movement. No presence. It had been standing 30 feet from me. It had been invisible to the camera at the same time. I filed my resignation that afternoon. On my way out I stopped at my department head's office to hand in my keycard. He looked at me for a long time. Then he said, "Did it let you see it? Not did you see something? Did it let you?" I did not answer. I walked out and I have not been back. I still have the field notes. The photographs. The plaster casts. The audio recordings of vocalizations that do not match any species in any reference library. And I have the biological samples. 3 labs refused to document them in writing. I have the samples and I have recordings of all 3 phone calls. What I found in the Appalachian forest has been there long enough to learn camera placement. Long enough to avoid documentation. Long enough that my department head knew exactly what question to ask when I resigned. It is not a new species. New species do not know how to avoid being recorded. New species do not maintain deliberate perimeters around 40 square miles of mountain terrain. New species do not choose when to be seen. Whatever it is, it has been in that forest long enough to understand us. Long enough to study us back. If you are a biologist, a ranger, a researcher, or anyone who has spent significant time in the Appalachian range and you have found something that came back inconclusive, tell me in the comments. Use a different name. I will understand why. Subscribe to the Midnight Files. Some species are undiscovered because we have not found them yet. Some are undiscovered because they have decided not to be found. Until they are ready.